morning, Group A. Welcome to join us also from the via the web. So we start with Rona's presentation first and continue discussion. Please.
Thank you. Okay. Good morning, everybody. I'll take my glasses off. So this is so to give you a little bit of context of what I'm going to be describing today. It's a genetic and cytogenetic family trio study of military veterans of historical nuclear testing. And we're, out of courtesy for our stakeholders, we don't release any information until we've shared anything, until we've published. And unfortunately, we haven't quite got the publications through yet. So what I want to be able to do today is, obviously I can't give you any results, but I want to be able to outline our approach. So, okay, so this is a bit of background. The UK undertook a series of atmospheric and experimental tests in the 1950s and 60s, and principally these took place in mainland Australia, Montebello Islands, and atmospheric tests at Christmas Island, present-day Kiritimati, and also there was an experimental program which released a lot of radioactivity into the surrounding area in Maralinga. And here's the hierarchy or the timeline of the operations that the British undertook. And the grapple series include grapples 1, 2, and 3, X, Y, and Z. And so there's a number of different tests within each of these operations. So the potential for exposure to ionizing radiation. So the atmospheric tests obviously release substantial amounts of radioactivity. It's released into the upper atmosphere, and it contributes to the global levels of radiation. And locally it can fall, it's fallout. And this could be earlier or delayed. If we just zoom down to here first, the majority of the personnel were deemed not to be in a high risk area or role, and indeed sort of witnessed the tests from miles quite distant to the actual tests. However, there were military personnel whose their job was to actually sample the radioactive cloud, and also the ground crew who cleaned these aircraft when they came back from the sampling are thought to be those that are highest risk for exposure. There was also a ship called the HMS Diana, and their task was to sail through the nuclear plumes. So there's known groups or of military veterans based on their role that are known to have had a higher potential for exposure. Additionally, those that were based at Maralinga during the experimental program, which did various different tests to look at the potential for explosion or for fire hazards, what would happen to source. And of course, that released a lot of radioactive material, and then the personnel involved in the cleanup. So the majority are not in a high-risk role, too far from ground zero, and no or negligible dose, but there was no record of internal dose measurements made. Okay, that's not moving. There we go. So concerns about the impact on health remain today, but they started in the 1970s, but adverse health effects in veterans and also in their children. And there was some epidemiology studies that were commissioned by the Ministry of Defence, and these studies here, and actually they've just been updated by Gillis and Haylock. whereby the most recent study shows that there is a small but measurable increase in mortality and cancer incidence in these individuals. There was also a health needs survey carried out, identifying what the concerns are within this community. Around about 2007, there was a study carried out by a New Zealand group where they did MFISH, which is 24 colour karyotyping of New Zealand test veterans, and they found a threefold elevation in translocations, which they associated with being present on these ships that were within the location of the test sites. There are some technical and some interpretation issues with this paper. However, the margin of the difference between the controls is quite striking, between the controls and the tests is quite striking. So that sort of reignited a lot of interest and the generation of this study, really, to better understand any genetic health consequences, and so looking for any evidence of historical exposure in veterans, and then any evidence of genetic changes in the first generation children, and then obviously to make this information accessible overall. So this is our design. We need to define our study population, go through all the approvals. This should say invitation, receipt, processing and analysis, because it was a huge logistical big project. And then we had our samples arriving into my lab, where we did the majority of all of the cytogenetic analysis, and then the whole genome sequence samples went for whole genome sequence analysis with Yuri Devrova's lab. And then each arm is looked at separately. Okay, so I want to be able to, I'll go into the details of the actual analysis in a few slides, but this is looking at who our study population are. So our target was to recruit 50 nuclear test veteran family trios and 50 military control veteran family trios. And we identified all of those through the cohort that was set up for the epidemiological study. So that's the UK Nuclear Test Veterans cohort. Inclusion needed to be registered with a UK GP, a doctor. Veterans born 1935 or later. It's actually, for the majority, it was 1937 or later, which made them 80 at the time of sampling. And they needed to have a wife or partner with a surviving biological child, all of whom were resident in the UK. We excluded those known to have cancer or any treatment that could interfere with the genetic analysis. So we had, the original cohort was of 22,000 test veteran men. So as I said, the majority, there was a concern about ethical approval, and actually this restricted our age for the majority of the sample to 80 or younger. And essentially that brings us down into these operations down here. That we wanted to get as many veterans that have been at multiple tests to increase the chance that they may have been exposed, and also to ensure that those that were at Maralinga were involved. And then the special groups that I mentioned earlier, they're actually defined as special groups by the Ministry of Defence. And so we wanted to include as many of those that we could. And so we extended out for the aircraft flight at Hanaklu and the HMS Diana to 82. So once we had that, we were basing it, we had a sort of ranking algorithm that was just purely based on number of operations, what the operation was, their special group status, and if there was a record of those. And then we identified controls, veterans matched on age, service and rank, and these veterans were all serving in the tropics, and so, confounding wise, exposed to similar vaccinations and so on and so forth. Okay, so this is just a summary of what is a huge analysis undertaking. So our first, the first question is there any evidence of historical exposure? We always keep a question mark here because clearly, as I probably should have pointed out, only 8% of, I think it was about 20% of the 22,000 were monitored with external dose badges, and 8% had a non-zero dose. Of that 8%, the vast majority were less than a millisievert. So, so the likelihood is that we would be not getting veterans that had any record of dose. So we wanted to use cytogenetics to use classic markers of exposure, so reciprocal translocations, but also given the potential for internalized contamination from long-lived plutonium, for instance, we wanted to see if there was any evidence of ongoing exposure, complex chromosome aberrations. So we used the technique of FISH. The second question is there any evidence of genetic alterations in first-generation children? So and these are all adult children now, average age of about 50. And so we've done a variety of different examinations. We've done G-band analysis on all of the adult children. This is looking just to see the constitution of what they're born with, looking for any obvious chromosome aberrations. And then we're looking for non-clonal aberrations using classic solid stain. So that's unstable chromosome and chromatid aberrations. And then this is the whole genome sequence analysis, whereby we're looking for SNVs, indels, structural variants, clustered mutations, in a very similar manner that the Jaeger paper described yesterday. So looking at so that's, and there's more details can be found on our website here. So I have taken out, I haven't taken out those numbers. Actually, I did change in an updated version, so they've not, this isn't the latest version. Okay, so this shows the number of cases that were supplied by us. So essentially, the number that were less than 82 thought to be alive and cancer-free that we got from the Public Health England, the UK cohort database. And then using our selection criteria, we made a long list for flagging, and then that went to NHS Digital, which is our UK registry to allow us to get GP contact details using their national insurance number. And by this time, we could see that there was a reduction, 25% reduction in those that were alive without cancer, and then also quite a number that clearly had emigrated, and so we've reduced here. We know that 405 veterans were invited because we received that information back from the GPs. We don't know how many were invited, but we don't have response, that they didn't tell us. And then we can see that the numbers that actually were eligible and were interviewed, that at each stage in the process, we lost, as it were, individuals. So, yeah, this is not the latest version that I provided. If just quickly, if that's all right, it's not that much different, but again, it's just because. No, do you know what? It's fine. Okay, I did. It had. Okay, thank you.
Just had a comment. The EMFISH slide, Rana, was from a female.
Oh, was it?
Sorry. What? Ooh.
I just have. I have. Oh gosh, do you know what? That's That just shows you. So I just picked the prettiest pictures that I just keep using. Thank you. I just, it's the same, but I just took out some numbers. Yeah, as you know, but I've had that one for years. Right. Yeah, that's it. So, so over 3,500 invitation packs were sent to GPs. And what I wanted just to show here is the sort of practical aspects of recruiting and consenting aged families. So you have here the eligible couples, and the recruitment started in 2018, and you can see that we went right the way up to over 2 years. And then you can see that the couples were consented quite quickly, but having adult children that are in their 50s, trying to get them consented, and then also, it was tricky. Basically, that required a lot of calling and a lot of additional work, because after this, the couple are consented, we ask for the first child that was conceived after returning from the last test site. So that reduces bias about who they're selecting in the family. And then we have to go through the GP and go all through the consent process as well. So then it's only when the whole family trio are consented that then a blood packs get sent out. And then they have to go to their GP to get the blood taken. So again, that is not easy for working adults. So this was receipt of our blood coming into the lab, and you can see that although we started to study here, we got the majority of the samples in the year before the shutdown. So we didn't manage to get all of those that were actually recruited for the study. We have gone back and contacted, but there's only so much contact you can do without actually, being too pushy to try and get the samples, and also GPs. It's actually difficult to get appointments. So I wanted just to go through the questionnaire. So for the military veterans, service and periods of service, location, what their role was, and there was free text as well as specific questions. And we also asked if there was any birth defects or genetic disorders that affected them or their children, and then the number and the date of birth of the children, the rest of the SIBs. And then we had a number of very specific questions, and these are questions that allowed us to verify both with the free text and with public health, what's in the Public Health England database. The potential other radiation exposures, we were very well aware that the motivation for a control military family, might be based on what their role was, and so there's issues with regards to that. So these were a variety of different, obviously, questions relating to confounders. So as I said at the beginning, there was only 20% monitored, about 8% had a non-zero dose. There were special groups that were defined, but we knew that we would not be getting a population that we had any record of dose for, or very limited numbers. So what we did was, when we had the questionnaire information and all that testimony, we could we developed a ranking system, so potential for exposure. So we wanted to assign veterans had higher potential for internal, external exposure. So everybody got assigned into the lowest rank. So that was equivalent to observing from a safety zone, or just being in the vicinity for 12 months on camps and so forth. And then they could only move into a higher rank if there was sufficient information in the testimony or their special group status that said that they had repeatedly gone into the forward or contaminated areas, had roles that were sampling or cleaning aircraft, either within days and then they were in the highest rank, or if it was 3 months after the test, such as like the cleanup workers and so on and so forth, then where the dose and the dose rate would be lower, they were in rank 2. So that was done independently of of any of the other analysis. So this is basically where we're at. So we have our questionnaire data that's allowed us to do various different verifications, develop our exposure rank, and collate all of our exposure and confounding variables. And we've described the paper and all of the information that we've got from the questionnaire in this paper here. And this is accepted, and we're waiting for proofs for to come through, and then we can share that. We then have our different arms of the analysis, as it were, and the whole genome sequence analysis. That was just done on 30 test veteran families and 30 control veteran families. So that is decoded, and that has been analysed. Group analysis comparing the germline mutation rate, incidence between the two groups. And this is, I'm just sending the revisions back for this now. So then we'll be able to present this one. So with the MFISH and the G-band, there, the majority of that analysis is finished, and we're undergoing the statistical evaluation, and we're also decoding parts of the instability data on the children. And what we're now able to do, if we think about all of these initial parts, we're describing them as separate, just entities. The germline mutation is just information as a group by comparison of the two groups. As we actually decode everything, we're in a position to evaluate not just between the groups, but within families across the different analyses. So everything from the questionnaire information through to the cytogenetics and obviously the germline. So I think as we move forward, we'll be able to do more, integrate across the different endpoints, essentially, which might be more, will offer more insight than just group analysis. So then, so the additional piece of work that we've added in is to collect urine to look for any evidence of plutonium in the veterans. So that urine is actually coming into the lab now, and we'll be doing a pilot study and then taking that further if need be. So that's where we're at. I wanted. So I just want to finish with this, really, is to say that this is some of the other research. This is how it fits into the research of at CHRC, Brunel, nuclear test veterans, low dose exposures. We're obviously working with other occupational and medical exposed individuals, and we're using very similar techniques to ask the various different questions there. But we're also doing in vitro studies looking at the multiplicity of exposures and doing a lot of sequence data mining to look for, understand sort of effects on neurological pathways. And this kind of links in a little bit with some of the psychosocial aspects. So there's anecdotally high levels of mental health issues, both in veterans and in descendants. And there's we've got a couple of psychosocial-based projects underway, and also health and well-being. And we have some sort of engagement. We're building our education and engagement projects up. If I can go back to this, I kind of wanted to finish with this. At this meeting, we've been hearing, about trio studies and thinking about what epidemiology, classic epidemiology is or isn't being able to inform on. And what's really interesting is that the number of trio studies that are being done, and thinking about future and how we've got past cohorts, and we talked yesterday about the difficulties with some of those cohorts, knowing whether they're definitely preconceptional or both in terms of their exposure timing. But I think, thinking moving forward, it's about standardizing methodologies and learning from the, how we've gone about things and how everyone's gone about things, so that we can actually improve in how we do these studies in the future. So it is about thinking about structured interviews, getting as much, involving psychological aspects, but having real detailed interviews and clinical, physical health and mental health aspects as well. I think we need to I think people are dealing with whether to include people with cancer and radiotherapy differently. We obviously excluded them, and we're excluding people that are at risk of being exposed. Other people might just have to consider those as confounders, but I think we need to address that for moving forward. And the main issue that I think that we're all finding is that people have different scans. We're all being scanned for a variety of, all these medical issues. Some people might have more scans because of underlying medical issues that might be relevant. But either way, when we've got very low, potentially, a low incidence of something that might be happening, we've got a lot of noise with confounders, mainly I think through medical exposures. I think we were talking yesterday about maybe panels of biological endpoints. So I don't think it's just about mutations and clustered mutations. I think we need to be working with genetic epidemiologists and really exploring the sequence data in a lot more depth. And also having samples so that we can do epigenetic endpoints as well. And then thinking about our methods to be able to really integrate across the endpoints, because we're not seeing one, or potentially what is anecdotally reported, it's not sort of individual health outcomes or specific ones. It's a whole range. And so to be able to measure and to see, to actually detect if, is there something there above the background? I think we need to be able to know how that we can integrate more, I don't know, statistical mathematical tests or something to be able to understand that a little bit more. And then I think, as Eric mentioned yesterday, and then with others, it's really how we share this information with stakeholders, and just thinking that moving forward, it's not these historical groups as well, it's medical exposures, workers in medical environments that I think are going to be coming through as a larger population. And that's me.
Thank you so much, Rona. Yes, sure. Let's discuss the paper.
So thank you. Very interesting presentation. I found a lot of similarities with our problems we have to deal with, but although the approaches are a little bit different. So my questions are related to dosimetry. The first one is simply for understanding. What's the point of collection of urine? Do you expect plutonium 70 years after the event?
So the question is, we're not entirely sure, but if there is plutonium, there's sensitive methods to be able to detect it. Maybe I've not answered that properly. Yes. So the Maralinga, particularly, very, very dirty, lots of radioactive plutonium, and the potential for internalized contamination, breathing it in. Yes, there's so it's not, it wouldn't be unexpected if there was internal contamination. Being able to detect it all these decades later might be difficult, but yes.
Okay, but from the point of view of metabolism, biokinetics, so it should not stay for 60 years in the body, except of, for instance, Chernobyl, there were few very unique cases when a hot particle was inhaled into lung, it was encapsulated there. But again, it has nothing with excretion or removal. Okay, so I understand that.
Yeah, so I think the plutonium does have a long. So yes, I think, what am I trying to say? Can someone help me? The bottom line is that plutonium would be incorporated, and the biokinetics of it is that there would be majority would be retained, and then there's a slow excretion through the urine that can be detected.
It's not your own invention, but my guess is that imagine what is the initial amount to be excreted in measurable amounts, say 60 years after. So I doubt. But anyway, so you have good dosimetries, I suppose. My next question is related to mFISH and your attempts to measure doses. So you mentioned that only a small fraction of people had measurable doses at the time of the test. There were not everyone had dosimeters, but among those who had dosimeters, dosimetry badges, the doses are pretty low, which is good. And I also assume that the dosimeters were distributed to the hottest categories. Those who are more safe, they didn't receive dosimeters, so their doses are presumably even lower. And then you base your dosimetry on mFISH. But mFISH is known to have pretty high sensitivity threshold, about, say, 250 milligray. You mentioned 1 milligray as some typical level for those who were monitored. So my question, do you see any doses among those for whom you took samples and made fish with dosimetric purpose?
So I can't share that with you just now, and clearly we've got the issue with age, and so any detection would need to be above a very high background. So we're not trying to do dosimetry based on mFISH. We're trying to see if there's any evidence of higher levels of aberrations that might be consistent with exposure. So, but you know, we know that the background rate of translocations, for instance, is very high at that age group. One aspect of using mFISH was to follow up on the observation from the New Zealand study that complex chromosome aberrations were detected more often. They didn't record the aberrations properly, but basically they said that they saw more complex aberrations in the test veterans compared to their controls. And complex aberrations are characteristic of high LET radiation. So the question was, could they be detected, could they be measurable, could they be measured, and could they be evidence of ongoing internalized exposure? So that was one of the key reasons for using mFISH as well. But it's not, this is a stakeholder driven project also, where there is a sense of lack of trust with understanding whether the dose records and is such. So partly it was to follow up in their interest in what had been done in the New Zealand study that seemed to show this very large difference in translocations.
Interesting way of discussion to given that had taken microphone. Okay, so I understand your point, and in this case, I may give some advice if you want. So I very much, I don't believe that you will find any plutonium now, but what you say about these New Zealandese subjects with high, presumably effect of high LET radiation. So indeed, it could happen that the people were wearing dosimeters. The dosimeters measure gamma and maybe beta, depends on type. Maybe at that time they were film-based dosimeters, not TLDs or OSL or whatever. So they record only external weakly ionizing radiation. At the same time, those people could inhale plutonium and got exposure at that time. And now you maybe don't see any traces of plutonium which was inhaled again, say 60, 70 years ago, but they leave the traces of exposure which can be detected with mFISH. So maybe it's an explanation of this discrepancy which is obvious to me. Okay, thank you.
This is good discussion, I think, and related also to the questions that we have for the group. So there are questions like which biological samples might be informative in epidemiological studies, and about the endpoints to be studied, and age of the people. At what age should the genetic or epigenetic effects be studied? These are elderly people here. And I also have a question about the cancer. You excluded those with cancer. Then again, somebody might think that, okay, perhaps those that have the cancer were the most exposed. Have you compared the doses of these, those that had cancer with those that didn't? Or some way tried to figure out if there's any relation to the dose?
So we wouldn't have any information because we wouldn't have received any veteran that had a record of cancer from Public Health England from their database. But is there, so in their recent update, yeah. So what we'd need to do is go back to the update, that's the Gillis and Haylock recent update of the the sort of long-term follow-up mortality and cancer incidence. And so in just this latest follow-up, for the first time they're showing that there's I think it's a 3% difference, so increase in cancer, 2% mortality, or the other way around. But, and then the subgroup analysis showed that there was higher, maybe the subgroups, the special groups, those that flew through the plumes, and obviously those that have a higher dose. I'd need to get that paper out and then I can answer to you, but we wouldn't be able to answer that question directly. That would be in that update. I'll send the paper to you.
Questions, please.
Yeah. So this is a great study. You know, but because we've been discussing this. One question is just out of curiosity. You have a Pito as co-author.
Julian.
This is Julian. Okay, is this the son of Richard Pito?
Brother.
Oh wow. Okay. Wow. Okay. And the second question is, you don't. Wouldn't it be interesting to have just a random control trio? I mean, if you imagine you find something in one of those exposure groups, but you don't really have dosimetry. So what do you conclude? Do you expect those response kind of?
Yeah, I mean, I think this comes back to the point I was making earlier about when we can talk through the results, the motivation of the military control groups, participating, maybe concerns about what they may have been exposed to, so on and so forth. So how representative it is of the general population. But I guess we haven't done it, and we're comparing overall frequencies with what's published, normal expected frequencies based on age, for at least for the cytogenetics. But no, we haven't. So I guess the answer is future studies should have a normal control trios in as well.
I think so, because you will be, imagine you find a slight elevation. Imagine you will be talking to these guys, and how will they take this? Because you have nothing to compare it against, and you say, well, we can compare it against historical data, but I mean, I don't have, I don't really have a good solution, but I suppose that including a control trio group would maybe be, I don't know. I'm just bringing this up as.
Yeah, continuing on that, so because they are 80 years old, so by age, your chromosomal aberrations should certainly increase. And there might be also this clonal hematopoiesis type of effects. So
So we can detect clones through the cytogenetics. So if they're there, we would be able to look at that. The flip side of this is these are 80-year-old, they might represent very healthy, surviving, and so there are, timing issues of when you do these studies. So
would it be good to include even individuals with cancer? Because you will have Andre asked about controls. This can be the positive control for chromosomal alterations, not necessarily. Yeah, not necessarily. No, not necessarily for dose or to relate that to radiation exposure, but from biological endpoint of view, if you have chromosomal alterations, translocations, then if you can relate that to cancer, unless if they are circulating in blood.
you're talking about classic translocations or something, but that would be easy to pick up. The issue is the radiotherapy and the chemotherapy, and yeah, but it's messy enough with the diagnostics and with the medical exposures. So actually, if you're looking for small numbers of low frequencies events, of how you actually make sense out of the data. So we've excluded it, but I think it's, there's clear limitations in the study. But I just think that moving forward, when we're I think for doing germline mutation, do you need the cytogenetics? So, and if you're if you're detecting the germline, the de novo mutations, then presumably it's the child that you would be not wanting to have cancer or radiotherapy. So that might make it, if you just restricted it to sequence analysis and didn't do any cytogenetic, I think that would be a way.
Do you know, you already mentioned diagnostics, but do you know anything about other sources of exposure? For example, variations in radon that they were exposed to?
No, nothing on radon.
Thinking about the future and the epigenetics studies, do you have any recommendations for samples or sampling, or what additional information might be needed?
I'm not. So, I mean, methylation. I mean, the obvious. Well, the talk yesterday was excellent, and he showed the 3 key changes. I actually don't know about the sample processing. I mean, I think it's very similar to that you get the blood, and then you just need to make sure that they're preserved in the right way to be able to do the analysis. But what is it that you want the study to do? Do you want the study to identify a mechanism, or do you want the study to identify if there is transgenerational adverse effects? And I think if it's mechanism, then you want to have the genetics and the epigenetics. If I'm not entirely sure, given the whole reversibility and everything, what the epigenetics will tell you about outcome. Again, it relates to a potential mechanism. I think the biggest thing is having the links to the clinical registries and actually relating all of this back to verifiable medical records, so follow up. I don't know about anybody else, but I think so. For me, the epigenetics is not about proving something's there. It's more about understanding mechanism, but I don't know, I might be wrong.
No, so when I think yesterday's your presentations and also on epigenetic talk, the whole genome sequencing data whether that data tells you something about alterations in factors involved in methylation or any other epigenetic mechanisms, because you can pick that out from the database, right? Um, not specifically looking for epigenetic markers, but you have the data of whole genome sequencing, and you can find the factors involved in any type of epigenetic pathways. Isn't it possible?
That's not my area of expertise, so I will not give an answer, but I also was thinking about, would this study provide a possibility to confirm or do not confirm the findings reported for the Chernobyl trio, when they saw some association, but it was for maternal gonadal dose. They didn't see for paternal, but if to see on the leukocyte telomere length in offspring in relation to the paternal gonadal exposure. I think it would be possible, and that also could be another informative piece that could come out of the study.
Yeah, we're actually planning. So there's subject to funding. We need to get grants written, but basically, yeah, there's sequence telomeres analysis that we can do, but there's also from the cytogenetics we could do the telomeres, and that's an obvious thing to use. But the other stuff, and I think we talked about it yesterday, is that these sequences, they're rich in information, and it's about collaborating with genetic epidemiologists, and actually that's what we're trying to set up, is maybe archive all of the sequences, the whole, all of the raw data, and then explore a whole range of different questions, because I don't think we're looking, yeah. So I don't know, I probably went off a bit off there, but
I have a technical question first, and then another one on epigenetics. What was your approach? I mean, you did also G-banding and MFISH. Did you screen with G-banding and then you analyzed in details with MFISH, or you did both? Both, and MFISH for all chromosomes, or did you have selected chromosomes analyzed? This is one question. And the other one is not only for you, but we are discussing now that it was investigating epigenetic changes as well. And then, okay, but if we want to be more concrete, what to investigate in investigating epigenetics, how to start, what should we study? Well, just an example for your study, but that can be extrapolated for other studies as well, because you also said you are planning to study epigenetics. How would you start?
so we don't actually have any plans to study epigenetics at this point, because I don't know if we'd have to see, we've got some blood samples and whether they've been stored in an appropriate way to be able to do that analysis. So, but the work that we want to move on to next is actually is maybe the telomere work, and then also the data mining, the sequence data of that we have, more than what we've already done, and look across, look at individual subgroups of families rather than the entire group. So so we don't have specific plans in this study at all for epigenetics. I just think moving forward for like medical occupational workers, and thinking about setting up prospective studies and how you might actually start to collect samples so that they can be used for these sorts of studies. That's where I was thinking about including epigenetics if for future samples. Your first question, MFISH was done for the veterans only, for the fathers. The G-band was done on the adult children. We've not done MFISH on the adult children. We're doing just solid stain block analysis on the adult children. So the G band for constitutional, and then solid stain for any genomic instability, essentially, to see if there's any evidence of that. Okay.
No. Okay, just, yeah.
Yeah. I have some concerns about, I mean, we have been discussing about telomere length data. Most of the telomere length measurements are done once, and the data is compared with the control population, and this will not give you any information about telomere length shortening. So, I mean, this is true for all telomere association studies, telomere length association with diseases, for example. One-time measurement of telomere length will not be useful unless it is substantiated with chromosomal fusion or aberrations, because there should be a functional consequence of telomere length changes. Okay, then telomere length changes occur over time. It's not immediate. Okay, it's not like dicentric formation. So you need progressive cell cycling. If the cells are stable, if the cells are quiescent, then you may not notice telomere shortening. If you notice telomere shortening, that should be chromosome fragment loss. Terminal regions are broken and eliminated. That in that way, maybe your average length will be less if some chromosome ends are lost. Okay, so I mean, whenever I review grants or even papers, I ask people to do prospective telomere length analysis. For exposed population, we do not have data on telomere length prior to radiation exposure. If you don't have that data, then it's very difficult to conclude anything from telomere length data per se. I mean, that's my concept, because I have been measuring telomeres for entire my life, and
Yeah, yeah, but
that's the problem. If you compare with different
yeah.
populations, then you see shortening or lengthening. But if you measure in one individual over 10 years, then if you see shortening, that is real.
Yeah, I mean, I understand that if you want to study the, let's say, the dynamics of shortening, but if you compare one group and another group, and the age is the same, and everything is the same, and in one group the telomeres are shorter. Yes, but if the group is large enough, you will get a picture. If there is, everything is difficult in life, yeah, but
The length ranges from 10 to 20 kilobase pairs. Okay. Then, if you see on an average, okay, 10 of us, we have 10, 15, 20, some may have 25, all right? Average, you will get 15. And if the other population if you get 12, then you say it is shortened. It's not true, actually. I have short telomeres, but that doesn't mean that I have some disease or something like that. So average length is very difficult. Average population, yeah, it is very. Sometimes it is misleading because.
Also want to just to ask the same question in relation to Andrzej's point. So if in the group that are of the same ethnic group of from coming the cases and the information coming from the same population, and if we do the dose response model adjusting for the age sex differences, you think it also one time measured telomere length will not be informative? So what's then, can I ask the question about the
I suspect because we have 46 chromosomes, right? And 92 ends. No, 46 into 4 ends, right? So each chromosome has its own length as well. So on an, I suspect, I mean, you can get a gross understanding of the telomere dynamics, but that should be reflected in physiological functions.
How you interpret the findings of the Chernobyl trio study when they showed
No, I see
assess the association between leukocyte telomere length in relation
No,
to
no,
the parental gonadal dose.
even for chromosomal dicentric data, we know that lymphocyte lifespan is important, right? How many times these lymphocytes have divided in vivo? Do we have that information? No, right? So how do you calculate the rate of shortening? Because telomere shortening occurs every time a cell divides. Okay, so how many times your stationary lymphocytes have divided, unless the primordial stem cells have eroded telomeres fast enough to be reflected in the lymphocytes, right? I mean, we have to take that into consideration. Biology is important.
Yeah, my question is, what is the hypothesis for radiation exposure?
Yeah.
Shortening the telomeres?
No, the same is true for when I see publications on radiation-induced senescence, for example. That also follows a different mechanism. You see, senescence is measured over time, not immediately after exposure. So there are a lot of publications on senescence, radiation-induced senescence, or telomere shortening. It's difficult to understand.
Yes, in fact, I remember in one of the EU projects that I coordinated, there was this study comparing radiation exposed and non-exposed group, and the hypothesis was that the telomeres would be shortening, but went the other way around. So I haven't seen it published yet, but
It's a Nobel Prize discovery.
yeah.
But I'm not
Yeah.
criticizing the data, because the data is important,
But
it should be followed up.
so I think that's a really important point and something for study design. But I don't think we can ignore thinking about telomere length as a useful marker of health, not of exposure, but thinking about it in the descendants, and it is thought of as a useful marker for health. Now, does that mean that needs to be monitored over time, or you could compare two groups? Because a lot of studies have compared two groups, and
No, I mean, why I'm bringing this point is there are telomere length has been associated with everything, even short-term unemployment. So then, occupational exam, stress. Yeah, no, that's why it's too many associations. So that's why the average population average may be a little bit difficult to comprehend. I mean, we need to be cautious about this, but it's a good endpoint to measure even something physiologically relevant.
Yeah, I think you're right with this high variability in the individual telomere lengths. And if you compare an individual telomere length to, I mean, two groups, then you can fall in this, what you explained. It might help if you compare individual telomeres to a group and not one group to the other, because then you might easier catch the difference that a particular individuum has telomere lengths slightly different from the average, or falls into the within the standard deviations, or within the confidence interval, but more at the limits. It's something similar with what we find with the various lymphocyte subpopulations, that it's a very huge variability within the population, and then you see that an individuum has a change within a particular lymphocyte subpopulation, still it falls within the normal limit. The individuum changed a lot, but due to the high standard deviations is within the normal limit. And then sometimes, not always, but sometimes helps that you compare an individuum's value with a group and not two groups. And then the other thing, which is more or less not very serious, but maybe we should also start thinking that because we fall into this problem that you have to compare values to a previous control value, but you don't have that. So yeah, we would invest in biobanking of healthy individuals to have a sample to which to compare. And for this, blood samples are maybe quite easy because quite a lot of people have blood samples prior to the disease and compare certain values, but obviously not cytogenetics and many others.
The astronaut study or cosmonaut study done by NASA, right? They may had the telomere length before they went for space, but that that's what that makes it difficult because yeah twin studies, right? I mean that doesn't make any physiology. No, so 1 minute. I want to suggest something about that. Okay, so when we don't have the telomere length data at different time points, but we have one question here: what tissue samples can be useful? So if in an individual you have lymphocyte data, if we can measure telomeres from some other organs, which is highly proliferative, for example, or non-proliferative. So then within an individual you can compare if any organ loses telomeres faster. I mean, this is even relevant for epigenetics. Okay.
Could you do the assay where you stimulate in vitro and compare? No, but then you'd go through a few cell cycles.
That way you measure the short-term rate, right? Maybe skin cells, for example, because length depends on telomerase activity.
Yeah, but that's if we're looking at telomeres, is the relevant thing their length at a given time point or the dynamics? Because it's two completely different things. Because you're suggesting monitoring them over time, but if you're looking at changes in length, it's a different type of marker than just length at any time point. And which would be the more informative? And I'm not an expert on telomeres, so I don't know. You think it's the dynamics?
Yeah, because even, I mean, if you understand the replicative senescence mechanism, once the cells start losing telomeres, right, and there is a point where either senescence or carcinogenesis will kick in, because at that point, when the telomeres are too critical, then you have either senescence, cells will die, or you have suppression mechanism will be mutated, then the cells may activate telomerase. That is not.
So you need to know where you are at any given time point and how fast you're approving this critical point.
But senescence is good because the cells will be not at the organism level, but at the cellular level. I think this is even relevant for epigenetics when we were discussing about epigenetics yesterday.
Right. So we are discussing preconceptional exposures and germ cells. So in fact, in these reprogramming stages, there is extension, I think, of telomeres. We should review this a bit more detailed. And well, if the telomeres in the parents are shortened, and they keep on shortening, there will be nothing left for the child. So they have to be extended at some point.
But I think that this is the question. For example, if you compare children of fathers who are, let's say, 80 and fathers that are 20, still the children have the same biological age. They start with 0. So yeah.
Yeah, I mean, they will not be exhausted anyway.
And
Till
when they
a-
start from the same endpoint, then having just one additional one afterwards, if you assume they started the same, there's already a big
Yeah,
information on
the
how
rate
fast they
rate
progress.
of shortening is very important because that will lead to some disease prevalence, for example, because that is more critical. That's why measurements at multiple time points will be very, very important. Yeah.
Unless you assume that to begin with they're all the same, then you already had time. You implicitly already have one time point.
So is this is still under question 2, what endpoints? So for me, genetic versus epigenetic, is it not both? Is that where we've come to rather than one or the other? Because it's not really, yeah. And
I'm not sure why the discussion was derailed to the telomeres, because in your studies,
Because I have
yeah, in your studies, it wasn't one of the, at least not major endpoint. If I may go back to your study, if I understood correctly, your selection criteria excluded the cancer patients, and after that, the F1 population was taken from the non-cancer veterans, I should say. Okay, so the selection criteria of non-cancer veterans is understood for the F0 population because of the exposure, radiotherapy exposure. But do you think there is a bias when you look at the F1 population? There is a bias, descendants of the healthier parents. So you're basically studying the F1 within the healthier general population. So it might underestimate the effect of exposure in a way. In F1, if you look just at F1, is it fair to say?
thanks, Andre. Yeah, I think the limitations are there that we made the decision to exclude those with cancer, so therefore these are healthier 80-year-olds. If the adult child who was the first child to be conceived after return from a test site, if they also had treatment for cancer, we couldn't include them. It was purely, it was related to measuring genetic changes being, the impact of radiotherapy on what we'd be looking at. So I think, so yes, we do not have those that have cancers. And I'm looking at, when I look at the Jaeger paper, there was 5 families who had cancer, and actually without doing the cytogenetics, that's why I was saying maybe moving forward to do the germline mutation, we don't need to think about excluding the cancers for the parents, but we probably would have to consider radiotherapy as a major confounder if it was in the child that was being sampled. And that's what we need to be talking about, because we haven't sampled those with cancer. I think.
Quick question. We are discussing this specific study, or we're really, yeah, that's why, the telomere length was in, because I think it's all related to the
Okay.
group, first bunch of questions. So it was, yeah.
I mean, this can be relevant for
Yeah.
questions are relevant for individual
Mhm.
studies or studies in general.
Okay, my next comment actually, yes, goes into the more general topic of endpoints, but from the study, I think the from the blood samples that you have in that study, it would be very difficult to do epigenetic measurements. Okay, they're more dynamic, and you need special processing for sequencing. It's cytosine, methylated cytosine conversion to cytosine, non-methylated and methylated. They have to be pre-processed and then sequenced. So if you haven't done that, from the sequencing results you have, it's impossible to derive any epigenetic information. Just technical note. But you mentioned there's some clinical measurements from the GPs of those patients. So, and I think that would be very, very informative and interesting. And it's a systems biology turn to the analysis of the results. For example, there is a NASA study that looked at the multi-omics and multi-modality of those twin study, and very nicely showing how from a multitude of different endpoints, including omics data, you can derive really relevant health-related information. So I think like maximizing the output from available biosamples, biobank samples, blood, maybe you can extract RNA, do transcriptome. There's questions about transcriptome, but still, and the clinical information from the GPs also, I think is useful.
But just a comment to the study of possible analysis of epigenetic effects. What about ATAC sequencing? That should be, that I think would be informative. It's I don't know if you're familiar with this technique. No. You're using the fact that you're using an enzyme that cuts chromatin that is open, and then it cuts, and then you can sequence this. So you know where it is open, and you know which sequences it is, broadly speaking. But I'm not just to flag this. But I have another question that maybe it just occurred to me. Maybe it's stupid, but we are brainstorming. In none of the studies have the lymphocytes been irradiated, and the response of the lymphocytes to radiation was studied. Would this be an idea to do this? I'm not so sure.
Well, one question about the is to see about radiosensitivity, not of obviously the veterans, but of the offspring. So and they're classic assays. So they could be built in as an endpoint, you know, and to identify maybe subpopulations more radiosensitive in the offspring.
I wouldn't use the term radiosensitive. I would say the response to radiation in terms of maybe impaired DNA repair, something like this. Not
Okay, but more
modified response to radiation, something like that.
No, and then the radiation biologist will ask whether you plan to do a dose response, because then a lot of other questions. I would prefer using single dose.
Yeah,
Focusing
the DNA.
Exactly. Yeah.
Okay, so going back to our questions, and the question one, if there is any evidence on existence of germ cell effects following local external irradiation or total body external radiation and whole body radionuclide accumulation. So, for example, the radiotherapy question, are there trio studies on radiotherapy patients? That would be.
Yes, there are studies on the childhood cancer survivors treated with radiotherapy and their offsprings. I cannot tell immediately by heart the size of the group that's been studied, but there are publications on the topic. So the only question is that it's absolutely clear to everyone that this population cancer, childhood cancer survivors, it's a special population that different from the other general populations exposed to occupational, environmental, other sources of radiation. But yeah, there are studies on. Yeah, yeah. That's true. But yeah, they got cancer as the case for this very good dosimetry. Yeah, but I can have a look, and for the I will just
as a control group for those studies, do they use cancer parents trios but without RT radiotherapy? So
the chemotherapy
yes.
that also
Is, so yes, the answer is yes. They have this control group to compare at least the radiotherapy with chemotherapy patients.
have a look. I don't want
Oh, okay.
confuse you. I will check.
Okay, okay. That would be interesting too. It would be different. That's why you could compare. Yes.
Yesterday you presented, apologies, you presented what I thought was, yeah, you didn't present any radiotherapy trio studies yesterday.
On purpose, because again, I thought we are more interested in general population, cancer-free population, let's say, because this is a really special topic, and I can have a look and give an update, to the group. Yeah, that's no problem.
Brothers and sisters of the patients.
I'm not aware of any studies like that.
So the question one, is that also from classic epidemiological, because we're obviously focusing on trio, but is, so I guess we need to bring in the update from the ABOM studies, and some of the studies that have shown weak, but some evidence of congenital abnormalities. That, because I think that, yes, we need to bring, so there's some evidence from the epidemiology. I think the whole genome sequence studies, what we heard yesterday from the two studies that, showed that there was no obvious germline mutations. So, but they hadn't actually linked any, done any sub-analysis of families that have reported health conditions.
In addition to congenital alterations, information on the unbearable pregnancy outcomes that also been presented.
Yeah.
You correct me if I'm not wrong. I'm just thinking about the parental exposure, and we always talk about the first conceived and live-born child. But what is in the period between after the parental exposure and first conception that didn't end with the live-born? And I know, again, from the Chernobyl accident, from the residents of the contaminated territories, women were even advised to terminate the pregnancy because of the risk of deliver a sick child. But definitely there is no any epidemiological data on that, and we cannot address it just speculation. So we don't really know about the fetuses or even maybe embryos. I don't know, depending on which gestational stage the pregnancy was terminated. So we have no any data that could give us some insights about the effects of parental exposure.
Okay, so to the best of my knowledge, it was not systematically studies in our study, but at the beginning it was a concern. So whether we will face the problem of these abortions and or delayed conceptions, but indeed we had plenty of children born very soon after the exposure. So at anecdotal level, as you call it, we don't see it. But it was no systematic study.
And so for like the military veterans, and I think same with the French and US, there's anecdotally in the population and surveys, which are fraught with bias, do highlight or do show a concern about increased stillbirths and and congenital abnormalities. The issue is that there was no record, so the birth registry national databases weren't up and running until I think the 70s, and then actually it was sort of localized. So there's no way of going back and doing proper epidemiological studies. And I think that's.
And I think that was similar in what we heard for the A-bomb survivors when none of the registries started until 3 years after. So for the very early years, I think in any of the epidemiological studies, there's very limited data.
Yeah. From Chernobyl studies, was there in the survey you have done, whether this question was asked to the parents, whether there was any misconceptions? Okay, so yeah, I mean that would add up to the in the questionnaire. Yeah. Yeah.
I apologize. In 4 minutes. The break is your business. My business is the scientific council of my research center, so I have to leave now. Or if break is. I just can tell you that I'm impressed by the discussion, so which is completely out of my area. But nevertheless, I learned a lot, and I think that it's a good area to study, but it is very early phase. It's my impression as an outsider. So maybe I'm wrong, but I'm very sincere. So anyway, so I hope it will end with some good result, and with the assistance of dosimeters, of course.
No, I have a follow-up question on the question you asked to Rona earlier, right? The plutonium accumulation, and how long you think plutonium can remain in the body?
We should look at the SRP publications on biokinetics. There are big tables. Actually, it's even in publication 103, I think, it's an amendment. But,
Yeah.
so its information is well known to those who are interested.
I mean, in Mayak group, yeah, exactly. Yeah, her, because they did measure even 40 years after exposure.
Part of my concern, which I didn't express, so to measure, so in our center we had lab which was measuring plutonium, which was different purpose, and I know that it's very difficult to measure plutonium because laboratories can being contaminated very soon, and the laboratory background affects a lot your sensitivity. So my question was unasked question: what is the sensitivity of your method?
I mean, maybe it is not urine
Um,
measuring urine, but they did biophysical estimation, bone.
So in our case, we made it was a big project. Feces were used because urine is considered not very stable because of individual variability. Feces is, but it was for a fresh plutonium inhaled.
So we're collaborating with specialists that do this. So the Norwegian laboratory will be doing the processing, and then it's getting atomic mass atomic mass spec in a specialist unit that does this routinely. We wouldn't be doing it. So yeah.
It's very specialized. I apologize.
Thank you for coming. So perhaps are there any other points for the question one? For example, are there any situations where radionuclides could be accumulated in gonads?
Soft tissues around. Yeah. So I'm trying. maybe we could ask Svetlana, but because I'm trying to sort of think about something I wrote a couple of years ago about justifying this, and I can't remember the details. But for uranium and plutonium, soft tissue and bones, but I specifically I don't think it's accumulation.
There was some discussion about cesium actually and the beta radiation. So if cesium, yeah, but beta is short ranged. Is there any possibility to higher doses or no?
In mouse studies, we don't see differences. At least in the mouse studies that we do, we don't see like the mice are fed with water containing cesium, and the distribution is homogeneous. Yes, same with tritium. But some organs have different sensitivities. So, but in terms of levels, I don't think it's there's evidence. And any comments from the virtual part of the group? Taking a break.
Yeah, possibly. They were having their coffee break.
Yeah, well, it's actually time for the coffee break, but and after the coffee break, I think I will go to the group C to discuss a bit the evolutionary aspects of epigenetic or transgenerational effects. But after yesterday, I wonder if you would like to have a joint discussion with the biota group for the thinking about the experimental arrangements or epigenetics or transgenerational effects seen in biota. There's more evidence there. If you like, it's just an option.
Yeah, why not? I think that would be why not? Yes, but I have another. Yeah, yeah, yes. It takes two to tango. Yes, but I have another suggestion maybe because we should come up with, I suppose, with some kind of draft of the review article. And I was thinking whether we could actually, these questions are very nice as questions to be asked in the review article. I don't know whether we should address each one of them because we need to really look, yeah, go into the literature and kind of sit down and think about this. But perhaps the strategy could be that we make a document with all these questions and then we divide the work among ourselves and see how we proceed. And then, because we need some kind of plan how to go on. Yeah.
Yeah, I think it is a good idea to start drafting the special issue and the papers that could be there. I think that in our report tomorrow, we should give some indication of our understanding now. But clearly we can say that where we need to dig it more and go to the literature.
I think we formulated more questions than answers.
Yes, that's
maybe,
right.
Maybe we can select out some of the questions for specifically for the review, right? Rather than, I mean, we can list out all of them, but we can select. Yeah. 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 Exactly. Reorganize.
So the other questions after question 8, because there was quite a few pages.
Yeah, we can maybe. Yeah.
And also translating what we know about mechanisms into relatively effective.
So we have two interests to show what is known by now, and then pointing out that we won't know where we need more material. Anyway, I think it's a good idea to start.
Yeah, yeah.
Start going through this. I like the idea.
I also would like to come back to, I mean, I was maybe criticizing you too much or fighting with you. That was not my intention yesterday. I want to come back because I woke up in the middle of the night. Oh, I have to say this. No, to come back to your. No. Come on. That's the aim. No joke. Look, the idea of which mutations or changes are relevant for detriment. This is a relevant question. So I think it should be tackled, but again, not in a way of saying, well, suggesting that only those, because we don't give any recommendations, but a chapter on deliberating on which ones could be, and maybe these are better, these are most, that is absolutely relevant, I think. So I just wanted to come back to this to say, well, it's a very good idea. It's just that not recommend, but say, well, we looked at this and but the advantages, disadvantages would be such and such. Again, state, map the situation.
Thank you for comment. Just maybe to finalize this, I think this is a good way to identify gaps, because if we have known mutations or epigenetic changes relevant to health outcomes, we say that it's known, or we say it's not known, and that's the avenue for exploration for research. So it's.
Yeah, I think actually there are quite a number of epigenetic congenital diseases nowadays that have been identified, and genomic imprinting we have known for a long time, but I think there is more information now on epigenetic mechanisms that used to be.
Yeah, and one thing I did want to bring in was that some of these questions, I'm not sure we're qualified to, in this room, and whether we have the right sort of genetics backgrounds, or whether for some of the questions we need to either not address or ask someone to come in.
It's okay to bring in additional expertise. It doesn't have to be limited to the people here right now.
I just wanted to make that.
Yeah, I think it would be useful to have some medical geneticist here as well, but like quite a number of other experts as well. DG is looking for those. Okay, but let's after the coffee break.
Yeah, I think
you will be chairing, but you will start. Yeah. Yeah. I go to the other room.
Okay, so Cisco is in the other room now, so we can start again. Brainstorming probably some of the questions we have listed here. I know that we have been discussing the questions on and off, all the different questions whenever there was an opportunity. I thought probably we can start, say, with question 5, for example, and then if we have any ideas or concepts on on say which clinical and lifestyle information may be important to control confounding or to provide insights into effect modifiers? Are they the same for genetic or epigenetic endpoints? Virginia, you want to ask Nori now, or if he's available? Probably it's.
Hello, Nakamura Sensei.
Yes.
No, opposite
I'm here.
here. Yeah. Professor Nakamura, we would like to ask your opinion about the epidemiological evidences coming on hereditary effects and inter and transgenerational effects coming from the childhood cancer survivors and their offspring studies based on the review you published in 2013. Could you please comment?
Yeah. Well, for any epidemiologic studies following individuals for many years, that is very difficult and expensive. So, well, in terms of trying to understand genetic effects, well, I personally feel that, well, thinking about some kinds of diseases which may show up late in our life, we may probably don't need to think about that. More important thing is some kinds of diseases which causes, say, handicapped life for many years. That means heritable effects of which appears in young ages. That is the most important. But obviously, such kind of disease is not very common. I mean, hereditary such disease. So that is the reason why I took a different way instead of trying to find epidemiological approach. Instead, to know how many human confirmed genetic diseases, which are probably caused by loss of one allele of the two, autosomal genes. And I look at the online Mendelian disease online version. And I had only looked at once. It's a long way to go. But I could find that there are at least about 500 such dominantly inherited genes, and most likely caused by loss of function of one allele. Well, I tell you, the complexity of dominant heritable diseases, that's really complicated. So even the same gene can end up with two different types of diseases. That's not rare, that's quite common, because depending on the location of the gene and the amino acid change. And in that respect, animal studies are not very extensive. Instead, human diseases line up. That is really impressive. So I'd like, if I am asked to do something to contribute to this meeting, I probably look back the OMIM list again and make a long list. Say, look at these genes. Those are mainly malformation, mental retardation, cells or such kind of group of diseases. Then, that's a map that I showed you in my presentation. And it's a G-banding map, so many genes are located in the G-band negative areas. So that is why some black bars don't have many genes, but white bars, bands have many such genes. I'm sorry, where I'm going. My point is, I try to know what the genetic consequences in the next generation of atomic bomb survivors. But I now realize that it is not easy to find even such a tiny evidence for it. Then I started to change my mind. Okay, let's start to think, why don't we find such evidence? And in my opinion, there are not many radiation biologists who spend time and effort to think about that. So I thought it may be worthwhile for me to do that. That's my current thinking.
Thank you. So I know that in A-bomb survivors, RARF and other groups have been doing the sequencing analysis, right?
Well, yesterday Eric Grant mentioned about the plan,
Yeah.
but Japanese guideline, governmental guideline says when we do genomic sequencing for local people to characterize their something, well, one should get good understanding, not only from the blood donors, but also for people around them. And in the case of atomic bomb survivors, we know that, people were discriminated in the past because they were exposed to radiation, and also because their children may be malformed. Such kind of rumor really created very sad circumstances. And I really don't like to see it again 70 years after the event. But no one really knows how will it be possible to educate people who might be going to discriminate people. It's ordinary people. So that must be a very difficult job. So, well, doing science, laboratory science is no problem now, but mutual understanding with scientists and local people, that is more difficult, or at least to me.
Yeah. Thank you. You got the answer?
Yeah.
Yeah, we will contact you when when we start writing up the review, if we need more references from your review published earlier. Yeah, thank you.
Well, anytime, you are welcome,
Yeah.
and I'm happy to join you anyhow.
Yeah. Thank you.
Yep.
Okay, so we continue on the question 5. Yeah. Yeah. So if you want to continue, we can continue with Yeah. Yeah, because I was hoping that Zanko will be here, so when he's here, we can discuss about epigenetic, right? Yeah.
Wait, because I don't know if we need him really. I mean, because I think from his lecture at least, and that is what I expect he would confirm, is that we need both. I mean, to me, it's quite obvious that we have to. We cannot just say there is nothing about epidemic or there is a lot about it. We have to look into it. So, how many generations? But we discussed this yesterday, and we said 3, maybe, possibly, just out of curiosity, because well, scientific curiosity, because we don't know. Yeah. Right.
And it could be different for the mothers and the fathers. So you might need more generations if you include the mother who is possibly still exposed post conception, whereas for the father it doesn't matter so much. So there's an asymmetry there. But
Okay, 3, yes, but okay, I wasn't actually thinking about. Of course, you're right. Yeah, but what about animal experiments?
So this is human.
Okay, yeah, sorry.
For 3, so what the state of the knowledge since 2007, I mean, there's been a couple of reviews done. We started a systematic review. It's been really challenging. We've put it to one side because of workload. But I'd like to pick it all up again. There's a lot of it still in here. So we've done a systematic review looking at this question again, and what we'll do is try and get this published in this next year. But we need to bring it up to date because actually we collected all the papers up to maybe a couple of years ago. So we'll probably have to update it again to account for the recent publications. But what we've done is, I don't know if you're aware of the OHAT approach. It's a toxicology approach. So it's, we've sampled like lots and lots of papers, probably much broader than other reviews have done, to try and capture as many human studies as possible and animal studies as possible. And then we've gone through and essentially assessed them all for risk of bias and so on and so forth. So it's actually a very big document. And then we come out at the end with just a few studies that we can do some sort of meta-analysis on. So as I said, I probably want to try and bring that up to date with recent papers and then publish that.
So I think yesterday we were discussing about the risk calculation. That is part of this question number 3. Risk calculation for heritable effects. You mentioned about the toxicological profile, how whether they calculate any risk.
We're not doing anything about risk calculations. It's purely a methodological, systematic approach to gather papers on a particular topic and then assess them for bias and quality, and then to pull those studies that can be pulled. It's not anything to do with risk calculations. It's just collecting papers.
Can we calculate the risk based on genetic or epigenetic endpoints?
I don't think we can, but I think an important paper to mention here is the studies in the ABOM survivors, where there's consistently been a weak indication that there is a risk, but not statistically significant, but it's very consistent. So can we ignore that because it's not significant, or should we pay attention because it's so consistent? And there is new material since 2007.
Yeah, I mean, if we assume that this effect is real, then of course you can calculate the risk because you have. I mean, they did this yesterday. They had a risk per gray. I don't remember the value, but But I think, yeah, as Fika said, and as Dominique said yesterday, I think also the question is, because until now, again, we assume there are no effects in humans, but just for the sake of precaution, we assume that there are, but we don't have any evidence. But maybe this has changed because we now have evidence.
Yeah, I mean, I think it has. I think the ABOM update is provided. Yeah, yeah, and I think that does change the overall framework or how we frame the work that's been presented over the past decades.
The question I had for I think your presentation as well as yeah, Vadim's or even ABOM survivors. So whether the mutations, whether it's genetic or epigenetic mutations, seen in the parents are reflected in the progeny or F1. Are they looking at the same mutations or any other types of mutations? I don't. I mean, I'm not familiar with the, because I know that in previous Yuri's data, he chooses only, yeah, he chooses only few mini satellites. Whether the mutations in only those mini satellites are analyzed in the progeny.
My understanding, they do the same for parents and offspring, and especially for the trio study that you mentioned first, they detect this de novo mutation and they look for this mutation in their offspring. So this is this the answer to your question? Yeah.
I just wanted to know whether the same mutations are transmitted.
Yeah.
So it's what's not in the genome that is new mutation that's detected in the
Yeah, while in Dubrova study, he was looking on the mutation rates in parents and offsprings in the same mini satellite loci. That's, yeah. Mhm.
But not in the new
No.
genome sequencing.
No. For Kazakhstan and for Techerio population, no, nothing like that. Mhm.
For the ABOM survivors, the new material does actually give an indication that it should be taken into account. Can we already say that for the trio studies? Are they giving enough information that we say there is something there? Hmm. Yeah. Yeah. Yeah.
There was only one study we mentioned yesterday with the three families of atomic bomb survivors. You can check the year it was published. It was also in Professor Nakamura presentation. He mentioned this three-family study of atomic bomb survivors. With not remember he showed the concordance between the findings in different studies in terms of transgeneration effects. And atomic bomb survivors, this three family didn't show anything, if I remember correctly.
So I think, there's no, yeah. So from the germline mutation stuff that's been presented thus far, there's no obvious evidence comparing a group or versus another group or a dose response. So no, yeah.
Some showed, some others did not. And again, the big question is the quality of the dose assessment in those studies when you look on the
Yeah.
association.
Yeah.
It's very few studies where they really have a good definitive, like atomic bomb survivors, like Chernobyl studies. The rest, again, Mayak, they have the data. We also can ask Svetlana, but they didn't publish yet. I don't know, maybe they do some analysis. Maybe we can just ask this question to Svetlana if she can give us an update of the. Svetlana, can you hear us?
Yes, I'm here.
Yeah. Could you hear the discussion we had and give us an update on the status of trio studies in Mayak workers cohort? Because we saw the recent papers describing in a very detailed way the database and the available information on trios. Are the analysis ongoing or planned, or just, could you please tell us about the status of this study?
Thank you for your question. This question is very difficult for epidemiological study because I agree with previous докладчики, reporters, and this question is very difficult for many reasons, such as control group, such as reliable randomization study group. And now we start our a large epidemiological study on basic of child registry in Ozyorsk, which includes offspring of Mayak workers cohort. And now we cannot see any reliable points about epidemiological following, because these observations is ongoing, and maybe to the next autumn we will give the first reliable results. Thank you.
Thank you. Yeah, yeah. I mean, not for risk calculation. Right? No, if the same mutations are not measured, then whether that the potential use on risk calculation.
I'm not clear why the focus on the same gene regions or mutations. The TRIO studies that are being done now are whole genome sequence, so that's coverage of the whole genome. The ones that have been done in the past were specific regions, mini satellites. So, but I think that in terms of answering that question, compared to the state of the knowledge in 2007, I think it's the health outcome stuff, the congenital update from the ABOM, and possibly a couple of other studies. I'm not sure the TRIO studies are going to be able to contribute at this point in time. But they could, I think, moving forward. No, not yet.
Okay, so we move on to the next question. This is more of epidemiological. Health endpoints determined for hereditary effects, background rates, statistical power, and detection limits.
Look at all outcomes, all changes, and then try to identify the ones that would be most relevant for health. I think that was roughly what we agreed on.
In terms of detriment, we also, if we have to think about the background rates, probably the cancer would be the best outcome to look at, that we can clearly have the comprehensive information on the background rates in various populations, when for the, let's say, cardiovascular or non-cancer diseases. I doubt that we could get any reliable background rates that needed for the study. Cancer probably is the, yeah, that's based on the existing disease registries in the worldwide.
Thank you. With regards to health outcome of for neonates and congenital abnormalities, there's only a certain amount that are live births. So and registries are now available. I don't know in every what countries, but in the UK and probably registries that assess. I don't know, is there stillbirth records? But there's definitely pregnancy outcome records, which were not there decades ago. So I'm now thinking either of new prospective populations, like medical occupational populations, but also second generation, the grandchildren, and possibly being able to go through records for congenital and stillbirth. So, but that has to be sort of like a time limit of how far we can go back because of those records. But to me, there's an awful lot of background rates we know about congenital abnormalities and health outcomes. So I think moving forward, we can do that as an outcome as well.
Yeah. So I mean, you pointed out that we can only focus on cancer, but no, because there are yeah. Mm. Yeah. Yeah. Mhm. Mm. When you pointed out that what could be the other endpoints we can consider? Yeah, yeah. Do you have any? Yeah, yeah. Any suggestions? When we are talking about low dose exposure. Yeah, yeah. What about what I mentioned, the in vitro radiation response? Yeah.
So this is specifically epidemiological studies in humans, which is why you're saying cancer, but we then which is good because of all the registries, all the data. But then you have confounding stuff. You have age, you have lifestyle, you have a lot more considerations there. If you include epidemiological gathering of pregnancy outcome, yes, you have some confounders, but you're not going to have the age and all the lifestyle factors. So both of those, I think, and then I think in the next, because I'm assuming this is just purely for epidemiological stuff.
Yeah, yeah. So I mean, our group is preconceptional exposure.
Yeah,
right?
but this is the epi questions.
So can we have the stillbirth as a?
Again, I think the best person to answer this question is Svetlana, because for example, Mayak workers cohort, who are under very systematic, what we learned yesterday, medical exams with all the health outcomes being recorded, and it was done within the same clinic that also helps to avoid questions about the different approaches for diagnostic and validation of the diagnosis. So, and this is information also about the health of the offspring born to Mayak workers. Maybe this is also the source of information to look at the more than just pregnancy outcomes, cancer, and other health outcomes. So that we can confirm with Svetlana.
Yeah, because the health endpoints and detrimental.
Yeah, that's
Svetlana.
Thank you for your trust. We will hope that we can expand this study, and we now we control our feasibility of this huge study, and I hope that we will get enough results for these important conclusions. Thank you for your trust.
Thank you. Thank you, Svetlana. Yeah. Yeah, I think it's very difficult to answer the statistical power and detection limits. We know that when we go into low doses, it's, we will have to expand the study sample size, right? Um, yeah. Not so critical, right? It's the consequence which is uh, yeah. Yeah, yeah. Hmm. Yeah. Okay. I think Rona, you touched upon the next point already when we were discussing the question 4. Which clinical and or lifestyle information may be important to control confounding, example, parental age, or to provide insights into the modifiers? Are they the same for genetic or epigenetic endpoints?
I can't comment on epigenetic, but I would assume that some stress or whatever that is would have changes. But clearly, medical scans, some of which are, relatively decent doses, as well as, paternal age. So maternal age is really important for just different chromosomal abnormalities, for instance, generally independent, irrespective of exposure. But paternal age is really relevant for germline mutations. So there's a strong association with increased mutations with parental age. And so I would be saying medical exposures and age and other occupational exposures.
Yeah, but it becomes complicated because, for example, if you look at studies on the risk of leukemia among children, then you have a whole bunch of stuff like social status. I don't know whether this can be considered or not. And actually, there was something in the presentation from Hiroshima about this as well, wasn't there? Yeah. But then alcohol consumption, smoking, these are the usual suspects.
How about ethnicity? Because we have populations that not only Europeans, but Kazakh or Tatars and Bashkirs. Do you think, I mean, for the epidemiological studies, it's also important to address and take this into account when you're looking at the cancer rates, because it's clearly has an effect. What about the epigenetics? Is it a factor that we should take into account and adjust our analysis? And also that, yeah, definitely the parental non-radiation exposures, especially due to pesticides, to other chemicals that, especially when we deal with the environmental exposure scenarios, that's another. And it was not shown talking about the possible effect modifiers, that time since exposure. In the TRIO study, they didn't, of Chernobyl cleanup workers and evacuees, they didn't see any effect of time since exposure. But it doesn't mean that there is no such effect or modification of effect by time since exposure. I think we can include some, how shall I say, not standard, but classical confounders and potential effect modifiers that we use in the radiation epi studies. So I'm happy to provide the detailed list. Yeah, again, we also saw aged exposure of parents, because we had a wide range of exposure. They were not exposed, all of the parents in the TRIO study, they were not exposed at age 20 plus. Some of among the evacuees, they were people exposed at the teenager time or even earlier, if I remember correctly. That also could make the difference.
That's an interesting point. This age-at exposure effect, is it both for males and females?
It could be, okay. To answer your question, it's not always seen, the effect of age exposure in the cancer analysis, but it's assumed that, yeah, the age exposure is a potential radiation radiation risk effect modifier, with the risk decreasing with increasing age at exposure. But it could be different in terms of acute, protracted exposure. So that's, but you want to see the sex or gender? Yes.
One thing that occurred to me now is that, well, we know that the risk of chromosomal aberrations or any genetic effects in children increases with the age of the mother. And I wonder, and I never thought about this, and maybe we can discuss, include this deliberation, whether the radiation effect is additive or multiplicative. And I have no idea. Actually, for cancer, yeah.
Yeah, I think one more lifestyle information probably can be considered is a diet. Diet is I mean, when I started my PhD work on in utero exposure, initially, we were just coming up with the animal house, and I realized that the animals fed with high fat diet behave differently to radiation exposure compared to the low fat diet. The response is totally different. I had to, remove all the data from one set of food in mice. It's very interesting, actually.
So all of this requires really structured interviews with, professional psychology, design, so you've got what you talked about earlier, maybe stress, or the sort of bringing the psychological aspect in, but also, if you're thinking about diet, what you're doing is you're adding extra layers of complexity to how informative and how structured that initial interview needs to be.
On the other hand, if you include ethnicity, this goes into this. But yeah.
No, this can actually diet if you have, some compound, say antioxidant, that may modulate the radiation. Right?
The question with diet actually, it's actually two questions: diet of parents and diet of the F1. For example, of both, yes, for both. For example, we're planning a new study. It's a mouse study, transgenerational effects, not intergenerational. The mice will be exposed to radiation, and F1, half of the mice will be put on high-fat diet, and one will be non-regular diet. So a high-fat diet will be as a kind of a co-exposure or challenge. So we have some changes, potentially we have some genetic changes in this F1, and then they might reveal themselves as health effects like metabolic alterations, or may not. But if you add some challenging exposure, you may actually detect the difference. So I think that's two questions. It's a parental co-exposure to confounding factors and also F1.
I don't know whether we can really call it as it's lifestyle. Yeah. 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 So I think both, even diet can influence it.
No, but in general, are the relevant factors, are they the same?
So I don't know. Apart from obesity, there's a very there's quite a lot of studies done with regards to epigenetic inheritance of obesity, isn't there? There's some sort of link there. But I'm probably speaking out of turn. I'm speaking headlines without knowing detail.
Also relevant to diet and epigenetics, the content of methionine in diet, it's a DNA methylation, right? So it's known from the mouse studies that by diet, adding methionine to the diet, you can change. And epigenetic changes, they're very volatile, so to say. They're very fluid. So if we have only one measurement in human population, it might not be indicative of anything. It might be just reflecting the situational diet stress situation. So we have to be careful with epigenetic studies. And if you want epigenetic information, we should include like multiple measurements, multiple endpoints. So diet, yes, is a factor, especially for the epigenetic measurements.
How far should we go back with diet information for parents?
How far you can go, I don't think, like from practical point of view, you just ask a question, what's your typical diet? Yes,
But this is you know, when you're thinking about some cohorts that are environmentally exposed, then the diet becomes you know, about exposure as well. It's not too many. Too much complexity.
I don't think this can be an exclusion criteria. Oh, we don't know anything about the diet and you're not included. That doesn't go that far, yeah.
This is something we need to know more about to reliably assess what's going on. So it's an important factor that I think we all agree on, should perhaps be considered more often.
Yeah, I think it's good to include diet as one of the questionnaires in most of the epidemiological studies, right?
Just a practical thing, maybe because you're an epidemiologist. Because I was applying for an ethical approval to study to get blood samples from control people. We wanted to do it's for biological dosimetry, irradiate the blood and score chromosomal aberration. Then I thought, oh, let me include all this information about diet and to see whether this influences. Oh, I had to drop this because it was not. I mean, to get this, I had problems to get this through the ethical approval. Oh, you're now interfering into personal life. Ooh, okay, forget it.
I mean, there are so many lifestyle and other confounding factors that I think we should restrict our focus on those confounding factors. Well, the clinical ones are more straightforward. There we can really identify the ones which really impact radiation response. On the other hand, the different lifestyle factors which are putatively affecting or whatever, they can be mentioned, but I think we should focus on those lifestyle factors which are proven to interfere with radiation response, because otherwise we just lose our focus. Well, yes, with an effect, but there are lots of lifestyle factors, and then we can increase the list depending on the particular population we are looking at, and we will not find an answer. We can raise the problem as a potential confounding factor, but without any proof. But I suggest to focus and to analyze more those lifestyle factors which are already known to interfere with these outcomes.
I think for epidemiological studies that makes sense. So you could also do fundamental studies where you're trying to investigate the new ones, but if you're yeah, so in this context, yes.
I think it may be really important because earlier Professor Nakamura was mentioning about how you are going to tell people and educate, the population on these aspects. Then we may have to come up with some um, other factors which may influence the effects, right? Andre highlighted the socio-economical status, for example. Right. To identify, yeah. Yeah. Hmm. Yeah. So, in essence, what we decide today is that parental age, sex, and then yeah, ethnicity, um, medical exposures, social factors. Yeah. Yeah, time since exposure. Yeah. Mhm.
Excuse me.
Yeah.
Excuse me. I think that as a confounding factor, we should consider the not only mother's age, it's a moment of childbirth or parents' age, uh, and also multiple pregnancy, parity of delivery, and gynecological anamnesis of mother, and maybe chronic somatic diseases of mothers and fathers. Maybe it is important.
Yeah. Complications. Yeah. Yeah. Statistics and
And the key thing is that we want to provide a structure for consistency, because what is always happening when you've got these multiple studies and you're trying to pull them and to do some analysis is that if they've all been designed slightly differently. So actually the key thing is to, maybe not all data points can be collected, but if there is a consistent categorization of what would be good to collect, if that makes sense. Yeah, because otherwise it becomes a bit of a mishmash.
so when you mentioned the medical information, which also covers existing medical conditions, right? Say metabolic diseases, for example. or endocrine disorders. Yeah, exactly.
Yeah, I was about to say, I mean, how practical is all of this? For new cohorts, and obviously MyAct, it's linked to registries and screening every 2 years, medical. But maybe do we want to say what our ambition is for if we were designing new studies, and what would be practical and achievable to collect from normal general practitioners, or
Ethically, is it feasible to get all this? Right? Yeah. 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 So, I mean, this is true for both. The last point in that question 5 is same for genetic and or for. We don't really know. Yeah. Hmm.
We are not likely to find exact information on radon level, but we may be able to detect if that particular individual lives in a high background level, because they don't know the background level, but they surely know that, I live in a, and that's an information.
So you could discern different categories rather than, if not actual dose.
Yeah, we were waiting for question 6. You showed that slide, right? Yeah.
One question I think we're actually missing here, and that's, are there any new cohorts that we should be thinking about establishing now? So, like, for example, you're looking at the UK veterans, they're now 80. With hindsight, you probably wish you'd started looking at them years ago. So are there any groups that we ideally should set up studies with now? For example, medical personnel.
well, so if we stick with the test veterans, then new cohorts, the French tests went on for longer, so they're younger. So there's them, and then there's maybe pooling, some families of interest. I think the occupational medical workers is an absolutely obvious one to be thinking of as a new cohort.
Medical diagnosed exposure of patients, especially pediatric patients, and the doses from the whole body scan would not be tiny, so that would be another prospective cohort too. Mm-hmm.
We may ask either John or Yoshi if they have anything. John or Yoshi, are you there?
Oh, yes. Yoshi is
Yeah,
here. Yeah, I'm still worried about what I should talk, And for me, it may be very useful to, construct the list of papers which has been published after 2007 regarding with the hereditary effect with the parameter of dose range or dose rate or confounding factor and number of subjects, age at conception, or endpoint of effect, hereditary effect, and so on. So this kind of list will help us what we should much more focus on to discuss. And okay, just free talk, okay, brainstorming. The new source of hereditary effect may be a 100K genome project in UK for rare diseases. They sequenced the genome of Torio families, and so and I hope, I expect that there may be some offspring from the father or mother who are exposed to ionizing radiation in medical situations, because the paper published in Nature recently indicate the 100 euro genome project for rare diseases indicate hypermutation in the offspring who are born from the father who are treated with chemotherapy. So if we dig into these database, I hope we can find some new finding of the genomic changes in the offspring after exposure to parental after a parental exposure to ionizing radiation. And also my impression on the new ICRP publication would be more relevant to the tissue weighting factor for gonad. And from the perspective of radiation protection, we always focus on the low dose and the low dose rate. And as far as we obtain from the publications, we noticed that the epi studies on people who are exposed to low dose or low dose rate have not identified an vivid increase in the mutation in the offspring after parental exposure to ionizing radiation. So now the tissue weighting factor of gonad is just 0.08. But so far we have not identified the vivid evidence of the hereditary effect after exposure. So I hope, the tissue weighting factor might be much lower than 0.8, 0.08, and so on. So in any case, if I have time, I would like to, make a publication list, which I published after 2007. And with this list, I'd like to discuss which paper is more reliable based on the number of subjects or accuracy of those or dose rate and blah, blah, blah. So, and yeah, actually the questions listed in this slides are very difficult for me to answer. All these questions are, evidence, not evidence-based. We have no evidence or little evidence for answer these questions, don't we? And Okay, for me, age is important to consider the risk of radiation. Say, the offspring from father at 40 years old have double mutation frequency, the offspring who are offspring of the father who are exposed to at 20 years old. 20 years lag increase the mutation frequency by twofold. The doubling dose of radiation in induction of genetic effect is 1 gray. This means 20 years aging is comparable to the 1 gray exposure. It's a huge dose. So age is, a tremendously very important confounding factor to consider the calculation of the genetic risk. And also, as you mentioned, the time between the irradiation and conception is also important. The childhood cancer patient who are exposed to high dose of radiation or cancer therapy has offspring of the childhood cancer patient have not indicated the evidence of increased chromosomal aberrations or some defect of some defect at birth or something like that. And as I am a biologist, spermatogonia has a tremendous capacity to repopulate. Iron irradiation decrease the number of spermatogonia cell after irradiation, but the remaining cell repopulate rapidly. This may indicate that most damaged spermatogonia have been eliminated after exposure, and healthy spermatogonia may survive, and these cells, I have no evidence, but my idea is that survived spermatogonia have less mutations, I hope. But the situation is different for oocyte. As you know, the number of oocyte decreases rapidly after birth. And oocyte have a little capacity to repopulate, as you know. The radiation effect persists, and the number of oocyte does not come to the unirradiated level. So from the viewpoint of biology, the spermatogonia and oocyte, the response of these cells are quite different. Maybe I have no knowledge about the error prone or error free repair systems in these spermatogonia and oocyte, but the evidence which may evidence of the repair system in the germ cells, error-free or error-prone, may add some speculations of the this estimation of genetic effect of ionizing radiation. Sorry, this my talk is not focused on the specific matter, but just my idea I provided. Okay.
Thank you. That's I think some of the points are very relevant, and we are almost touching upon the question 6, right? Are there stages in spermatogenesis and oogenesis that are more prone to radiation-induced effects, whether it's genetic or epigenetic? And Yoshi has highlighted, I think, an important issue in that biology of germ cells. Yeah, thank you, Yoshi.
Okay. In addition, okay, the epigenetic effect. Okay, the Nature Journal this week has published a paper entitled "Maternal Inheritance of Glucose Intolerance via Oocyte TET3 Insufficiency." TET3 is the enzyme that they methylate the DNA cytosine in the zygote. And, as Andy discussed, the maternal conditions such as food with high fat may, change the outcome of the offspring. And this paper clearly indicates that, the gestational diabetes, mother who have not yet, conception, but have some conditions of diabetes, that mother's oocyte have decreased activity of TET3 enzyme. So mother condition may affect the epigenetic systems in this paper, TET3 enzyme. So mother condition is also an important confounding factor when we consider about the epigenetic effect of ionizing radiation. But so far we have no data regarding with these epigenetic changes in radiation biology.
Yeah.
So so we need a lot to do research. Yeah.
Thank you. I think maybe when you have time, you can send us the link to those papers which you have highlighted. I think it will be very useful. Professor Nakamura, you have a question, comment?
Dr. Shima's made me remind one thing. Current whole genome sequencing technique, which is called next generation. But to my understanding, this technique is good at detecting base change type or small rearrangement, but not very good at detecting deletions. And my concern is not the single base changes that may be transmitted to the next generation following an exposure to radiation, but deletions that may cause dominant diseases in the next generation. So my suggestion is I like to ask someone to look to find specialists in the genome sequence technology and ask him or them to evaluate what the current whole genome sequence data is that really valid, or still we may wait for longer sequence technologies that are better to detect deletions. That I happen to be a co-author of the Chernobyl's survivors whole genome sequence paper, but I'm always wondering that point. And I asked Dr. what's the name? I'm sorry, the head of this strategic group. How do you think? What do you think that deletion is that okay? And his answer was, they made effort to sequence depth 80 times on the average. So 80x is quite a large number because usually people sequence 30 times on the average or even less. So, but I'm still wondering technical validity of the current methodology. Thank you.
Yes, I agree with Nori's opinion. So we need the long read sequencing technique for the Torio studies, I think. And classically, we use RACGH for detection of large deletions, but it's not, a whole genome in detail, a whole genome analysis in detail. So there's some, frustration for using RACGH. But recent whole genome sequence indicate that, some large or small deletions and multi-site mutations is associated with RNA radiation. It's a kind of a signature of RNA radiation exposure. It is suggested. Do you agree, Nori Sensei?
That, well, the paper is still limited.
Exactly.
And
yes.
and well, that's one possibility. But multi-site mutation, that means multiple base changes within a short sequence, right?
Right. Yes.
The number of such events is not large. It's a kind of rare changes. So
right.
even, well, the result may be statistically significant, still it's arguable. And that is not very detrimental when we think that coding sequence of gene products is quite a small fraction of the whole genome, like 1 or 2%. So small changes, probably over 90% of such changes would cause nothing. But deletion is quite different. And in the 2001 UNSCARE reports, they are concerned about so-called deletion syndromes that include multiple genes, and curiously, they repeat de novo and repeatedly occur at some regions in the genome. So I'm guessing there may be some special sequence homology or structural similarity, whatever, but we are still too young to know why it happens. I mean, immature. I'm not young.
Yeah, thank you. Thank you very much. I think it was very, informative and interesting points you have raised. Yeah. Yeah. Okay. So I think we are into question 7, right? Q6.
Since we're still on the topic of epidemiological studies, most of the epi studies didn't start until years after exposure. So they're not going to give you a lot of information to answer Q6. So for the existing studies, I don't think we can see anything.
Okay. We move to the next question. 7. At what age should the genetic or epigenetic effects be studied? I think we touched upon this earlier and also yesterday. And what is our consensus on this?
Molecular, genetic, and epigenetic alterations, changes, endpoints you can measure in newborns. But if you want to follow up with, for example, cancer, disease, so it's a lifetime, lifespan follow up. So it depends on there's no clear answer. It depends on the endpoint, I think.
How long we can?
As long as you can.
Extend.
As long as you have money
As long as we find something.
Perhaps for the congenital malformations, it depends on the severity also. So what exactly the endpoint? So if you have a severe malformations that impact the function and the lifespan, they can be detected early on, and those there are like 2 to 3% of the newborns, and then an additional 2 to 3% of newborns have less severe malformations that will manifest by age 5, and in addition then there would be like 6 to 15% of mild transformation that you can see later in life with some small functional disturbance.
Definitely during the first 10 years of, right? Yeah. I mean, even other changes, I think it could be detected at least during the child development. Yeah.
Presentation from Zdenka, this cord blood could be the first approach, and it's easy to collect. I mean, if you set up the study when you want to assess the epigenetic changes in the newborn, so that would be just like first observation, and with the several repeated ones during the life course. But yeah, the I also understood that it's a lot we can learn from this what's the name of this screening at the birth. It has a special name, this test, that done to every neonate, that's the newborn. Yeah, exactly. Yeah, just like yeah. I need to find, but this is the data that also could be available for in case of the future. I will find it.
Yeah, I think the first 10 years of child growth, probably like our controlled animal experiments, right? There's not much changes going on, and beyond 10, then all the other factors come into effect, right? Is that because teenage?
Because of aging.
I think ideally you would also like to repeat the measurements, because I know, for example, for childhood leukemia, a lot of newborns actually have clones of pre-leukemic cells, which then later disappear again. So if you just look at birth, you may get incorrect estimates for effects.
So move to question 8. Is it the complete question? Yeah. Which biological samples might be informative in epidemiological studies? It's an open-ended question, right? I mean, mostly you collect blood, right? What else? And yeah, you collected urine. Urine is for a specific purpose, not for biological endpoint, right? Here.
I'm trying to make it relevant to sort of potential cohorts moving forward. And obviously, what we're looking for is plutonium, long-lived radionuclides. So thinking about medical exposures, they're not, so the targeted alpha therapy, all of that, they're short lived. So I'm not quite clear of what the potential for need to be able to do some sort of urine analysis moving forward.
Do we think that this is something where it's meaningful if we answer the question, or would this be something that would be so study dependent that there is no generic answer? Or do we want to make a recommendation to make it easier to combine studies?
I think going back to question one, we have questions about whether there's internal or external, what the knowledge is. But what I'm trying to think about, do we, so collecting other samples, it would be about trying to identify maybe radionuclide contamination, but what scenarios would that might be? So accidents, terrorism, moving forward, I'm trying to think of exposure scenarios that we would be seeing long-lived or relatively long-lived that could have a relevance. I may be wrong about medical exposures, internal radionuclide therapy, but I think it's days, isn't it? That
Saliva. Yeah, saliva can be informative. You have something.
Are we talking about parents or children or both? Because with children, with children, what is done in genetic counseling is that you take also skin samples because you want to know whether there's mosaicism or not. And if there is mosaicism, then that means it's not transferred from the parents because you would have to, you would expect to have, but I don't know if this is doable. I mean, because collecting blood is easy, but collecting skin is not so difficult either, but it's more painful. Those that have done it, like me, know.
Yeah, I think this is very much related to also the ethical aspects. So what you are allowed to do, and you have to have a plan what you do with these samples. So it's not usually accepted to just collect samples. Except for biobanks, actually, they are collecting samples for any potential future use, but that has to be defined later on and approved.
Well, they go to dentists, right? So they can collect some gum tissue if it's like serious procedure, or just exfoliated cells. So that could be from the dental gum tissue. You can extract even stem cells and do some in vitro ex vivo experiments, like sensitivity, for example. Expose tissues and then examine the kinetics of DNA repair. Difficult. Just that's why I'm saying just another idea to consider. If there is I don't know, you can submit a PhD project and get a special mini satellite project on that.
Yossi, please.
Oh, thank you. In addition to biological samples useful for the examination of genetic effect, as I mentioned before, is it possible to use the 100 Kilo Genome Project in UK for rare diseases? The Nature Journal last month published the paper entitled Genetic and Chemotherapeutic Influences on Germline Hypermutations. And this paper utilized the cohort comprising parent offspring torios. And these torios are exome sequenced from developmental disorder studies. So among these cohort, I hope parental exposure to another radiation torio may be included. I'm not sure, but that these kind of sequence database may be sometime useful. So yeah, that's my comment.
I think it's a really useful point. I'm working with a genetic epidemiologist, and we're looking at all the different databases that are there, particular questions. But we did look in the database that you mentioned, the 100 Genome Project, for radiation exposure, and the information is really limited. I mean, we will go back and look at it again, but it was maybe a couple of years ago. And the actual level of detail, there might be sort of cancer patients noted, but there wasn't sufficient detail at that time to allow us to be able to make any meaningful tests. But it's a really useful point, and I think moving forward, having databases, repositories of all the sequence data is really important.
Thank you.
And now that we are discussing preconceptional exposure, I think we should discuss also germ cells. So if we think about humans, of course, could be sperm, which is collected. Oocytes are very much more difficult. But I don't think it's feasible really for epidemiological studies, but perhaps some more experimental studies to look into, let's say, epigenetic markers in sperm, and if the histone markers are removed by the protamine and that kind of things. So, and if indeed, let's say, in vitro exposure of sperm is manifested in any of these epigenetic signals or something like that, that could be done. But I think that otherwise it's very, very difficult.
Okay, I think we covered most of the questions from epidemiological studies in humans, right? And time is also up, another 5 minutes. Yeah. If you have anything which covers all the questions we have raised here, we can just wrap it up for 5 minutes. If anything, last points to be highlighted. Dimitri? No. He's thinking or looking at the time.
I'm just thinking, are we going to discuss anything on the animal side, experimental, later on? Okay, so yes, I just a general thought that some results from human studies, you have some lead mutations or epigenetic alterations which you see increases. You can then take this information and design animal experiments to actually test hypotheses. You can formulate and test hypotheses. You can use genetic manipulation experiments. So I think it's it's important to keep the two domains, human and lab animal experiments, kind of talking to each other at least, and have this perspective in mind. And in also maybe for the report, for the review, highlight this. It's important, the communication between researchers in the two domains. Yes. Yeah. That's all I wanted to say.
I think we had a very good discussion, and we involved Yoshi and Nori, Professor Nakamura, and also Svetlana, chipped in for some of the discussions. I don't know what John's background is biology or John Matthews. Epidemiologist. Okay, yeah, but he didn't. Okay, I guess we can.
Yeah. Thank you for the discussions, and we break for lunch, and perhaps after lunch we bring the group C here to discuss the animal. Yeah, I think that would be nice.
How many are in group 3?
It's only 2 on site, so they can come here. We fit together. Yes, we adopt them. Yes, but many more via web. And they are,
Okay.
yes.
Maybe they can also join in. Right? Okay, I think that was a wonderful discussion. Thank you very much, and we meet again at 2:15, right? Yeah. Thank you. Thank you, Yoshi, Nori, and Svetlana. We will be back by 2:15, Budapest time.
Thank you.
Yeah, Budapest time, not
Sure.
Yeah. Thank you. See you later.
See you later.
Yeah.
See you.